Well, good day, everybody. This is Chris back again with the Ancient Scholar, and this is video 2 in a series of videos on receptors. In this video, what I want to do is I want to talk about some of the major or broad categories of receptors that we can run into. And then there are two major categories of receptors, and we'll just go ahead and write these down. So the first broad category of a receptor is what's known as an ionotropic, an ionotropic receptor, and then the second category is what's called a metabotropic. An ionotropic and a metabotropic receptor. And the way that I kind of differentiate these, in my mind at least, is when I think of ionotropic, I think of an ion. Okay, I think of an ion, and of course ions are positive or negatively charged substances in the body, particularly positive and negatively charged either elements or what we call polyatomic elements, and these can include things like sodium, for example, and the sodium ion in the body takes on a +1 charge. It's charged, it's lost an electron. Potassium is another type of ion, takes on a +1 charge. Chloride, we know chloride is chlorine that has gained an electron and it takes a -1 charge. A more complex ion, what we call a polyatomic ion, which is an ion that has more than just one atom, because sodium, potassium, and chloride are just involve one atom, ionizing one atom, but maybe something like bicarbonate or HCO3 negative one, where this is a more complex ion because it has a hydrogen, has a carbon, and 3 oxygens in it. But these are all ions. And so when I think about an ionotropic, I think of a receptor that has to do with the movement, okay? The movement of ions into or out of the cell, okay, of this cell. An ionotropic receptor has to do with the movement of ions, and I just think of you have ion actually built in to the name of the receptor type itself. The other type of receptor, the metabotropic receptor, I just kind of think of the term metabolism, and I just think of stuff, lots of stuff going on. So I have, rather than just an ion flowing in or out of the cell, what I have associated with that receptor is I have a cascade. Okay, I have a cascade of events. That occur. And I'm going to give you guys some examples, and that'll make intuitively a bit more sense. But these are the two major classes of receptors that I want to talk about, the ionotropic and the metabotropic. And when we talk about these receptors, there are two subclasses that are critically important. And the subclass of ionotropic receptors includes something known as Ligand-Gated Ion Channel. Okay, a ligand-gated ion channel. Now, a ligand-gated ion channel is nothing more than a type of ionotropic receptor. Okay, and then the very special class of metabotropic receptor is what's known as a G protein coupled receptor. Okay, G protein coupled receptor is a type of metabotropic receptor. Okay, so ionotropic and metabotropic are a bit more broad, and then subtypes include the ligand-gated ion channel, which is a type of ionotropic receptor, and then the G protein coupled receptor, which is a type of metabotropic receptor. So let's kind of now just look at the ligand-gated the ligand-gated ion channel. Okay, when I say something is a ligand-gated ion channel, what am I saying? Well, imagine that you have the cell membrane here, and I have a receptor, okay, and it is ionotropic, okay, because we know ligand-gated ion channels are ionotropic receptors. And let's just say that this receptor here, when it is open, it allows an ion chloride. When this receptor is activated and opens, it allows an ion chloride to enter the cell, okay? Chloride enters the cell, okay? So an ion is moving, so that makes it ionotropic. And what we mean by ligand-gated is a substance, okay? So that's my substance there, and maybe there's a part of this receptor here that has the right shape for that substance. That substance can attach to that shape. And the substance that attaches and interacts with the receptor here, it can interact and kind of form kind of what we call a coordination complex there. This smaller substance here that can attach to the receptor is what's called a ligand. Okay, that's really all ligand is. It's a little thing that it can attach to this receptor. You may remember from chemistry when we talked about ligands and metals, that's a situation where I have my metal atom, okay, so I have my metal atom here, and then I have these, in the metal atom we model as being, having this positive charge to it, and then I have these negatively charged ions that associate themselves with the metal, okay, and we call these negatively charged guys out here ligands. Ligands, okay. It's the same sort of thing that we have occurring here. It's just we have some substance, and in a lot of cases that substance can be a drug or even a neurotransmitter that acts as a ligand, and that ligand interacts with the receptor. And in this case, what's going to happen is this ligand is going to open this receptor. The ion channel will open up, and it will allow chloride to enter the cell. And a good example of this action is something in the central nervous system known as a GABA, G-A-B-A, that stands for Gamma-Aminobutyric acid, which is actually a neurotransmitter in the central nervous system called GABA, and GABA can act as a ligand. Imagine that this is my GABA receptor here. GABA can act as a ligand, can interact with the GABA receptor, cause the GABA receptor to open up, and when that does, that GABA receptor allows chloride to enter the neuron, okay? And when chloride enters the neuron, it makes the charge inside of the neuron more negative, and that action, of course, depresses the neuron and makes it more difficult for that neuron to depolarize. So GABA actually causes CNS depression. Okay, it depresses the central nervous system, and we have a whole class of medications called benzodiazepines, or benzos, that include things like Valium, Ativan, Midazolam, Xanax, etc. And these all work by interacting with GABA receptors or possibly by increasing levels of GABA in the central nervous system, depressing the neurons in the central nervous system and causing lethargy, causing sedation, in some cases causing amnesia, or causing you to forget things. So that's an example of a ligand-gated ion channel. It's just a ligand, a substance like a drug or a neurotransmitter can attach, the channel opens up, and it allows some sort of ion to flow either into or out of the cell. Now, a metabotropic receptor is a bit a bit more complicated, and what I'm going to do is I am going to give you an example of a G protein coupled receptor, because we know that a G protein coupled receptor is a metabotropic receptor. So what happens is I have some substance, okay, that can act upon that receptor. And when that does, when it acts upon, what we call agonize, in this case, when I agonize a metabotropic receptor, I activate. Agonize means to activate, antagonize means to block. In this case, we're talking about a substance agonizing the receptor. Instead of ions flowing in, what happens is I have different processes occurring within the cytoplasmic membrane or within the cell itself. In this case, I'm going to choose a very specific type of G protein coupled response because it is very relevant to the biochemistry of medicine. So in this case, what happens is this receptor will activate something called a G protein in the cell membrane. The G protein then will activate another protein called adenyl cyclase. And in biochemistry, whenever anything ends with an ASE, whenever I see that at the end, ASE often means enzyme. ASE, I just associate that with enzyme. And of course, enzymes are proteins typically that catalyze or make chemical reactions occur more efficiently by decreasing what's known as the activation energy for the specific reaction we're talking about. Well, adenyl cyclase catalyzes an intracellular reaction, and that reaction involves adenosine triphosphate, or ATP, and it involves a conversion of ATP into a secondary messenger molecule called cyclic 3'5' adenosine monophosphate, or simply CAMP for short. So this is an example of a G protein coupled response where I have a substance acts upon a metabotropic receptor, that in turn acts upon a G protein, the G protein acts upon adenyl cyclase, and adenyl cyclase is an enzyme that catalyzes the conversion of adenosine triphosphate into CAMP. And a good example of a G protein coupled receptor is something known as a beta-2 receptor. And the beta-2 receptor, we actually have them throughout our body, but we have them in high concentrations within our lungs is one specific area where we have high concentrations of these receptors. And in this case, when a substance acts upon that beta-2 receptor and this G protein coupled response occurs, the CAMP that's produced as a result of a beta-2 receptor being activated in the lungs actually causes dilation of the bronchi, or we call bronchiodilation, and we have medications such as albuterol and Xopenex, terbutaline, and so on. And these medications mimic hormones and neurotransmitters. They mimic the biochemistry of them, and they like albuterol and that can interact with these beta-2 receptors and cause this response, which leads to bronchodilation. So that's just an example of a G protein coupled receptor, and of course a G protein coupled receptor is nothing more than an metabotropic receptor. So there we have an example of ionotropic, specifically a ligand-gated ion channel, and then a metabotropic receptor, specifically talking about the G protein coupled response. Okay guys, I'm going to cut it off here. As always, thanks for hanging in there.